This is the first workshop of the genomic sequencing workshop series that we aim to enhance capacity building of researchers at international and national levels in terms of genomic sequencing for tropical diseases. And in this first workshop, we are focusing on field data collection and also apply genomic sequencing at field-based approach. So we are going to have experts and participants from many countries around the world who come and share experience and share their knowledge to each other in terms of data collection in wildlife, and also at the same time doing the lab test to identify pathogens for using genomic sequencing.
We did several workshops on training, mostly on disease ecology. So mostly people that are used to work with wildlife and want to interact. But this is the first time that we gather people that work also in the laboratory in the genomics. So we make the connection, we make the link between them. For people like us that collect the samples, we don't know where they are going. We want to know how the people want, what they want process, what kind of data they want from the field in terms of genomic quality. And the same, people from the genomics, from the lab, they will know exactly how the samples are coming there. This is the real big strength of our workshop.
As we continue our long-term observatory project to sampling different types of samples from rodents, bats and birds. So we gather the people in the field who are working on the ecology, biodiversity, and also at the same time disease ecology. And this is good opportunity when we can put also the other sector on the laboratory, which is like the people on the genomic studies like nanopore sequencing, next generation sequencing, how we can merge them together on the field sector and also laboratory sector to work together in 5 days workshops.
Right. We have been surveying for bat here in Muka for 3-4 years until now, and at the moment we totally capture bat in this area for 14 species. We need to survey more in the forest and also add another effort in the open space, like using bat detector or mist net more to cover the microhabitat like water body or the open space that can, in the future, we can cover the good number and representative of each group of bat in Muka. And at the end of the day, we can set up the database and the knowledge of the bat diversity and population here for the next generation of the student who probably come and study bat and help us monitoring the bat population in Muka using some kind of basic equipment like hub trap, or even using just only bat detector to do the acoustic monitoring, and then come up with the long-term monitoring program and attract more funding, attract more graduate students to keep coming and do research in Muka.
Right now we are Department of Medical Entomology. We are the part of the workshop in the vector-borne disease. So here we have the experiment and demonstration of the mosquito collection and some application for the research and also the surveillance system. So this is the increasing emerging new emerging disease. So I think it is good for the researcher and also for the public health that can prepare what is the new emerging that's coming to us. So I think it is useful and it will be benefit for the trainee.
Mosquitoes are the vector of the many diseases such as malaria, dengue, hemorrhagic fever, chikungunya, and Zika virus. Mosquito tapping and identification of the species is one part of this workshop. When we know about the correct species, it is very important because you can study further about the epidemiology, vector surveillance, vector control, and disease transmission.
We've been teaching DNA sequencing, and that's something that's very valuable for young scientists to learn about in this part of Thailand, where we have forest right next to this campus, and we're able to sample animals and so mammals and other vertebrates, insects and ticks, and able to look inside these animals and vectors for pathogens, things that can cause diseases. So what we're learning here is how to detect through DNA sequencing what is inside these samples.
I'm very grateful to be able to come here to interact with the researchers here. I think it's very important to share some of my experience that I have in my lab in terms of bioinformatic data analysis, and hopefully what I've shared in this workshop will be helpful for the researchers when they do the analysis.
So in this workshop, I learned a lot about the challenges of doing science in the field, and especially all the different parts of organizing a field expedition and collecting samples in the wild. It was not something I had been exposed to before, so to me it was extremely instructive. And also spending time in the lab, seeing actual experts do their thing, and taking the time to explain to us each step of the ONT process. That was extremely instructive, and I'm very grateful for it.
Mostly what I learned was how to sample mostly rodents, bats and ectoparasites. I mean, I never done it before, so it was very interesting and challenging as well. And I liked the thing that it was very practical compared to other workshops. And I learned also a lot about ONT technology. I heard about it before, but I never had the opportunities to test it, so it was very interesting and challenging as well.
So what I've learned from this workshop is all the part about the genomics that I've never done before. It was really also interesting to combine the fieldwork and the genomics after to see how the two parts can be combined together. Yeah, I think before this workshop, the genomic was also such a specific field, and I didn't really understand how it can apply to other ecological science. For example, disease ecology, but also taxonomy. And so yeah, this was the main discovery.
Currently, I'm working at Institute Pasteur of Laos, where we're working on research and diagnosis on emerging infectious and vector-borne disease. I learned a lot. I learned many things from this workshop, especially like genomic sequencing technology, like a nanopore that I have never dealt with. I hope that it will lead me to apply to my job, to get more understanding further beyond our studying right now. And the most impressive thing that I got is the new friendship from other participants and learn experience from scientists and experts in different fields.
For me, I had the opportunity to join this training, and I learned a lot from the speaker and from the team in the MUCA. Actually, I'm from the lab side, and I don't have many experience from the field work. So this workshop gave me a lot of experience and new knowledge about how to collect sample from the field work, how to store the sample with the good quality to do experiment in lab.
I'm working at the Pasteur Institute Cambodia and in the virology unit. What I learned a lot of from here, so like from the field and lab or data too. For example, like the field, I learned like how to set up the mosquito trap, rodent trap, and bat trap. For the lab, I can learn some from the yesterday, like the nanopore sequencing.
This is very interesting for me because this is a very new thing for me. So I learned a lot of things from here about how to catch the factors and how to process it and how to get data from that. I learned a lot of things from here. So I will recommend this course for other clinicians, not only for like ecologists, but also clinicians to learn more things so you can like explore more new things.
it is a great honor of our team to have a chance to attend this workshop. We learned a lot from genomic sequencing at the sort of spillover event, and we got a lot of introductions and suggestions from experts in the field, and we really appreciate this opportunity. We agree with the workshop team that it is very important to perform sequencing quite real time at the source of spillover, because we can receive information and actionable data because we are Armed Forces Research Institute. So our mission is to acquire real-time data and inform commander or host nations, military groups, as well as the public health authority. And we hope that the data that we provide, they will use the data to plan preventative measures or even policy to protect people from infectious disease. So very glad to be here. Thank you.
I'd like to thank the organisers for inviting me here. It's been very informative. It's not my area. I've learned a great deal of things about sample collection, how these samples come, and then eventually get into the lab. The benefit this is, in this institution, there's a vast resource that potentially can be tapped to provide new insights into how infectious disease is spread, both within animals and from animals into the human population. It's an enormous resource that hopefully will be made available to scientists both in Thailand and outside of Thailand in forms of collaboration.
So, from what we started, I think this is a good opportunity for any of you. Either you are normal guys, or you are a student, or you are nature lover. You can come and join us the workshop and learn from biodiversity survey, learn from experts on each group of animals and wildlife in Muka, and then share the knowledge with us, and maybe use the knowledge that we share in this workshop, and then spread the same knowledge to your group or your research community.
In part of the medical entomologists, I think it is very important. So here is a good workshop that is waiting for you for the next year. I think it is good news, good friends, and good relationship. Welcome and see you in the next year.
What we have to do is to prepare the next generation of researchers. And really want to do, and we discussed really that with Ajahn Kittipong, really, and with the help of Mahidol University, really to develop a field courses, but integrate field courses for the master, PhD, and even postdoctoral students, which merge this, and we can really complete on this.
We still have 3 more courses or workshops in our series. So the next workshop, we will focusing on data application, data visualization, like how to use genomic sequencing that we have in the lab and do visualization, to utilize those genomic sequencings that let people, like public health people, to understand the results and use the results for disease surveillance. And the third workshop will be about human genomic. So we will be focusing on bioinformatics, looking at the analysis of genomic sequencing of human part. And the last workshop, we will focusing on genomic sequencing and also bioinformatics that focusing on malaria protozoa. So yeah, we're looking forward for you to join our other workshops.